Hey folks, welcome back to NC State Dendrology. Today's lecture, we're going to shift away from angiosperms. We spent a couple of weeks talking about angiosperms, and we're going to look at gymnosperms, specifically conifers, which is the focus of dendrology. And so these are the last two lectures that you'll have before the second exam. So let's look quickly at the lecture outline. We're going to talk a little bit about the evolution of vascular plants. Just a little bit of review from early in the semester, specifically focusing on gymnosperms, and within gymnosperms, we'll focus on conifers. And so this first short lecture will be about conifer morphology, specifically foliage. And then I have a second lecture where we'll talk about cones and different types as we look at different families and genera in the gymnosperms. And you've seen a few of these in class. So the last few weeks we've been focusing on flowering plants, and now we're going to move to gymnosperms. So gymnosperms, what are they? They're vascular plants. So if you look at this sort of dendrogram on the right, you can see that all of these plants have arisen from an ancestral green alga. And then the most primitive of plants were the moths and hornworts, and then from that we have a branch for first vascular plants. So the first plants that had vascular tissue, xylem and phloem. And so within that we have our ferns, but then we have first seed plants developing here. And then from there we develop into conifers, which are more primitive than flowering plants or angiosperms. So we have a lot of conifers and gymnosperms in the world today, 1,000 species. And in fact, the textbook of dendrology, which is an old textbook, has it at about 770 species. So our knowledge is growing, and we're recognizing and distinguishing differences among different families and genera. So when we're talking about gymnosperms, we're focusing on seed plants that bear naked seeds. And so what we mean by naked seeds, these are ovules that are exposed to pollination. So they aren't enclosed in an ovary like we have with angiosperms. And like I said, these are more primitive than angiosperms. They developed around 350 million years ago as the first seed plants, and actually reached their peak of diversity during the Mesozoic era, so about 250 million years ago. And you can see some examples of gymnosperms shown on the right. So there are quite a few that we will not get to in dendrology class, but gymnosperms are fairly simple. We have 12 plant families, 88 genera, and about 1,000 species, as I mentioned. So within that, we can look at conifers, or cone-bearing species. Here we have the very vast majority of gymnosperms, 600 species. This is also the majority of species we'll learn in dendrology. We've already learned ginkgo in lab that, as I mentioned, was a really cool living fossil, right? Just one species that is still living today, but used to be a much more widespread genus, family, and class. And then we're not going to talk about palms in this class at all. Unfortunately, we don't have a lot of examples to look at. These are confined to the tropics, and we have about 300 species in there, specifically a genus called Zamia. And then we have Ignetum, which is a genus, also a tropical genus with about 100 species. So we're going to focus mainly on conifers, and then of course we had to talk about ginkgo. And so for this course in dendrology, we're going to look specifically at the Pinaceae family and the Cupressaceae. And we've learned examples of species in each of these families so far. So of course, Pinaceae includes Pinus, also Tsuga, Pseudotsuga, Picea and Abies, and then Cupressaceae includes Chamaecyparis, Juniperus, Metasequoia, Sequoiadendron, Taxodium, and Thuya. And then of course I included ginkgo at the bottom, even though it's not technically a conifer. So first we're going to talk a little bit about foliage and how to distinguish this. And this is important because if you travel around the United States or the world, you want to be able to recognize maybe not the species, but you can at least figure out what family and maybe the genus based on the appearance of the family. And there's a lot of evergreen coniferous trees that are certainly out west and other parts of the world. And so it's important to know the attributes of each of the genera. So Pinus is one that we'll focus on quite a lot. We've already learned Pinus taeta, or loblolly pine in lab, and the leaves, right, in this case, leaves are acicular, so they're needle-like leaves. And if you pick up a little bunch of needles that are on the ground under a pine tree, you'll notice that they are grouped together in bundles called fascicles. And the buds are actually, so remember, we always know that leaves are attached below buds. The buds are actually inside of that fascicle and usually aborted. So if you pick it apart, you probably won't be able to find a bud for pine trees. But what's important is the number of needles per fascicle is diagnostic. It can tell you or help you lead to an identification. So quick review from lab. How many needles per fascicle does Pinus taeta have? How many needles per fascicle does Pinus strobus have? And I think we'll learn about Pinus echinata and Pinus virginiata later in the semester. But Pinus taeta has always 3 needles per fascicle, so that's a helpful identification character. And that'll help you tell it apart from Pinus strobus, which has 5 needles and 5s. Pinus echinata is a little bit intermediate between the two, needles and 2s and 3s, whereas Pinus virginiana has a little bit shorter foliage and the needles are twisted in 2s. And so you can see this diagram on the right that we have the needles. These are the foliage leaves. And then these scaly leaves are the fascicles that those little bundles are contained in. And they're contained on a dwarf shoot. So looking at pine just a little bit closer, I just wanted to show you this photograph so you can see quickly these little bundles, right? So it might be hard to find a bud, but you can think of the needles or leaves as being attached in fascicles. So counting the number of needles per fascicle can be really helpful in trying to make a positive identification. We also have, so in the same family, Pinaceae, we have Tsuga, or hemlock, which we learned in lab last week. And hemlock has short, single flat needles. So you can see the surface area, right? They look similar, like they're pointy and acicular, but they're actually flat. And maybe you would call those linear or oblong leaf shape. And if you look closely at this photo on the right, you can see there is a small petiole that attaches those leaves to the branches. And then also a good close-up, if you didn't have a lens with you in lab, to see those rows of white stomata under the undersides of the leaves. So we're actually looking at the underside of a Tsuga branch, and you can see those rows of, that white is actually rows of white stomata. So those are the openings that help with gas and water exchange. Also in the Pinaceae, we'll learn the genus Picea. Picea is the spruce genus. And in this case, they have thick kind of angular needles, and they're borne singly on small bumps or sterigmata. And actually the singular of that, so this is a sterigma here that I'm pointing out. So you can see these little pigs. And a really important ID character, we'll talk about this more in lab, is that spruce is sharp. And you can see at the ends of these branches, those needles come to a pretty sharp point. And that's something you can feel if you grab a branch to feel it. North Carolina, we have red spruce, and we'll learn about red spruce a little bit more in lecture as well as lab. That's Picea rubens. And we also learn Norway spruce on campus where it is planted. Moving on to fir. So spruce and fir are the high elevations, the species found at the highest elevations in North Carolina. And if you look at the close-up of the needles for ABs or fir, you'll see that they're blunt tipped, right? So they're a little bit squared off at the end, so not sharp like spruce. So you can remember the mnemonic that fir is flat or friendly. You can grab a branch and it won't prick you, while spruce is sharp. And you can maybe get a glimpse of this from the photo. It also, ABs also has rows of white stomata on the underside of the needles. You're not going to mix it up with hemlock, I don't think, because you can see the kind of bushy bottle brush arrangement of needles on ABs, whereas spruce or hemlock has 2 rank, right, for the Eastern or Carolina hemlock, where the needles come out on either side of the twig. And even the Carolina hemlock is slightly less bushy than this ABs here. You'll also find those 2 in different habitats, right? With ABs being found at the highest elevations, Carolina hemlock down on the lower slopes. We have a genus that we won't be able to see in person, but larix or larch. And one really cool fun fact about this species is that it is deciduous. So those little bundles of needles will actually turn golden, as you can see on the right, and be dropped in the fall. And those needles are born in clusters on short spur shoots. And you can see there are many needles in each shoot. So it's a little bit different than a fascicle, but that's larch or larix. And then moving on to the other major family in gymnosperms is Cupressaceae. That includes Chamaecyparis, Juniperus, Metasequoia, Sequoia dendron, Taxodium, and Thuya. And I think that includes all the genera that we'll learn in this course. So one important distinction here and how you can tell Cupressaceae from Pinaceae is that the bark on mature trees is fibrous and shreddy. We will see that in lab with several examples. And then the foliage, which is kind of our focus for today, a lot of the species has flat and scale-like leaves. So you can see that here on the right side, you can see those little scales, right? Those are the leaves, and they're flattened and oppressed on the branches. Other conifers in the Cupressaceae have small branchlets and needles. So you can see in this lower photo, right, that's an entire, what they call it a branchlet, but it's really a kind of compound leaf. And then we'll talk a little bit more about cones in the next lecture. So an example from lab this week coming, Metasequoia and Taxodium are both deciduous. So we always think of kind of associate conifers with being evergreen, but that is not always the case. So we have Metasequoia and Taxodium, which are both deciduous. And so this was a very short lecture, but I think it's important to just wrap up really quick with gymnosperms being our oldest seed plants, one group of vascular plants. And then they were most important and diverse in the Mesozoic era, so 100 to 250 million years ago. They're still very widespread and doing quite well. So I wouldn't say they're in decline, but they've reached their maximum diversity in the Mesozoic. Remember, and it's important, gymnosperms have naked seeds. So you can never talk about the fruit of a gymnosperm because it doesn't have fruit. And there are more than 1,000 species of gymnosperms. Main groups of gymnosperms are conifers, cycads, nedums, and ginkgo. And then you're going to notice in dendrology, within the conifers, we focus on Pinaceae and Cupressaceae in this course. And we talk about foliage for the genera in each of these families. So that's important because when you may not know exactly what the species is, but you should be able to, knowing this kind of basics will help you identify at a genus level when you travel, and then you can grab a local field guide to see what the species really is and get it down to the species level. So that's just a very quick intro to conifers, and next we'll talk more about cones.